Komodo Island is a very area, plenty of animals and plants that coexist in harmony and peace, except for the Komodo dragon. Komodo dragon saliva contains several deadly bacteria that are able to infect their prey, even large animals such as in the picture. This characteristic is unique for the varanus, and scientists still don't know how are they able to survive with so many dangerous bacteria in their mouths. Due to the fact that the Komodo dragon's genome is still unknown, we are going to use comparative genomics to find the antimicrobial resistance that confers Komodo dragon's deadly bites. First of all, we are going to make a small introduction about Komodo dragons, and then we are going to present our materials and methods to use comparative genomics. Then some economic considerations, and we are going to find a conclusion to our project and some references. Varanus komodoensis, also known as Komodo dragons, are the world's largest lizard with long tails, strong and agile necks, and sturdy limbs. Komodo dragon eat almost any kind of meat. Generally, they bite its prey and let the bacteria and venom in its saliva kill the prey within a few days. After the animal dies, which can take up for 2 days, the Komodo uses its powerful sense of smell to locate the body. Around 50 different bacterial strains, at least 7 of which are highly selective, have been found in the saliva. Researchers have also documented a venom gland in the dragon's lower jaw. In addition to the harmful bacteria, the venom prevents the blood from clotting, which causes massive blood loss and induces shock. In the most common bacteria found in their mouths, we can find aerobic gram-positive and Enterobacteriaceae. The genome of the Komodo dragon has not been sequenced yet. There is no evidence of bacterial-resistant genes in their genome. Therefore, specific genes that encode proteins for bacterial resistance in Komodo dragons are going to be searched. Comparative genomics studies the relationship of genome structure and function across different biological species or strains by genome comparison with the aim of discovering how selection has acted upon these adaptations. The overall strategy that we are going to follow consists on a nucleic acid extraction. Then, their genome will be sequenced de novo following a whole genome shotgun strategy, trying to achieve chromosome or scaffold level. It will be assembled and annotated, and once it is prepared, it will be compared with other species so as to find out which is the genetic cause of their antimicrobial resistance. Exome sequencing will also be performed. For this study, a male Komodo dragon from the Zoological Park of Barcelona will be selected for nucleic acid extraction. In order to locate the genes that are responsible for the antimicrobial resistance, the genome of closely related species will be compared with the Komodo dragon genome. 2 varanids, Varanus salvator and Varanus exanthematicus, will be selected since they are similar to the Komodo dragon and without antimicrobial resistance. These varanids were initially thought to have bacteria in their mouths, but it was discovered that they didn't and might have a venom gland instead. There are no other organisms that present antimicrobial resistance. Therefore, it's not possible to compare it with other species that are more different genetically and present immunity against bacteria. Finally, reptiles without antimicrobial resistance, such as Anolis carolinensis, a common green lizard in America, or Crocodylus porosus, the saltwater crocodile, are going to be used as reference to map the varanus since their genome has been already sequenced and studied. Genomic DNA and RNA will be extracted and purified using all-prep DNA RNA mini-kit from QIAGEN. This kit was selected because it has a quick and simple protocol and allows the extraction of genomic DNA and total RNA from the same biological sample. The estimated amount of DNA and RNA will be around 40 to 70 picograms of nucleic acid per milliliter of sample. The concentration will be calculated using qubit quantification device. The C-value found for the Komodo dragon in the Animal Genome Size database was 1.93 picograms. So the genome size will be of 1,887 megabase pairs. The C-value was obtained by flow cytometry and it corresponds to the amount of DNA found in haploid genome, measured in picograms or million base pairs. The repetitive sequence of Komodo dragon was estimated observing information of genomes of Anolis carolinensis and other crocodiles, and it is around 30%. Since the other two varanids that were selected has not been sequenced yet, they will be also sequenced the same way as the Varanus komodensis. Their C values are 2.03 and 168 picograms respectively, and were obtained using static self-rourometry. The aim of this project is to achieve a chromosome level in a de novo genome sequencing. Therefore, 100 coverage is going to be needed. Also, for the transcriptome, 150 coverage was needed. The Illumina sequencing procedure is going to be followed. First of all, library with pair ends with around 300 base pairs per insert site and mate pairs for long reads libraries between 7 and 15 kilobase insert sizes are going to be constructed. Sequencing is going to be performed using HiSeq 3000-4000 platform for the pair ends and Illumina HiSeq high output V4 for the mate pairs. Then BioNano will be used in order to assemble and map the reads, reaching the chromosome level. These are the strategies selected for each kind of library. The size of Komodo dragon's exome is still unknown. Therefore, we calculated approximately using Anolis carolinensis as reference. Since their exome is 3.64% of its genome, the calculated exome size for Komodo dragon is around 66 megabase pairs. The RNA extracted, a reverse transcription will be performed with superscript 3 reverse transcriptase from Thermo Fisher, using random primers to obtain the complementary DNA. Once the complementary DNA is obtained, it will be sequenced with Illumina MiSeq V2. Gene-level fragments per kilobase of transcript per million map reads and differential gene expression are going to be calculated using Trinity software, a method that assembles transcript sequences from the novo Illumina RNA-seq data. Then the genome of all the sequenced varanids will be annotated structurally and functionality in order to locate all the possible genes and obtain the maximum information possible. To annotate, the automated genome annotation pipeline maker will be used. This pipeline is easily trainable and useful to annotate new genomes. Firstly, the repetitive sequence will be masked using the software Repeat Masker, which will detect the repetitive sequences in the genome and hide them, because if not, they will difficult the process of annotation with many false gene predictions. Then, the RFAM database will be used in order to detect non-coding RNA that would also distort the annotation. Moreover, gene predictors Augustus and Eugene will be used to detect potential genes. The genomes will be compared through BLAST with Anolis carolinensis and Crocodile's porusus in order to obtain a better annotation. The transcriptome data will also be used to identify the genes. Finally, to annotate functionally, BLAST-P will be used to identify genes and domains present in the varanid genome. Besides, the KIC database will be used to detect whether certain genes are involved in a certain metabolic route, and the gene ontology database will be used in order to obtain information about the different present genes. To evaluate the genome assembly quality and the annotation completeness of the Komodo dragon genome, the benchmarking universal single copy orthologous, also called BUSCO, pipeline will be used. BUSCO provides quantitative measures for the assessment of genome assembly, gene set, and transcriptome completeness, based on evolutionary informed expectations of gene content from near universal single copy orthologs, which is very useful for large-scale phylogenetic studies. Once the genome assembly is evaluated, it will be possible to know if the draft genome is a good candidate for genome annotation and subsequent analysis, as it has a similar gene space completeness as observed in other eukaryotic genome projects. Once the genome is completely annotated, it will be uploaded to the NCBI database.
A phylogenetic tree is going to be constructed to estimate the genetic distance between the species used in this study. The phylogeny is going to be assembled using the whole genomes of the species used. Mr. Bayes' program will be used to run a Bayesian phylogenetic analysis to obtain the tree. The genomes of Varanus salvator and Varanus exanthematicus will be compared with Varanus komodoensis in order to identify what makes them different from the Komodo. In other words, the reason why the Komodo dragon is resistant to the bacteria in his mouth, but other varanids are not. The main focus of this project will be the study of differences in the genes of these varanids, taking into account that phenotypic innovations usually evolve from gene duplications. It could be possible that the genetic difference was not located in a coding region but in a regulatory one. However, this is less likely. Gene duplications, losses, or mutations responsible for this special characteristic will be searched. Genes related with the immune system, salivary gland, and mouth of the Komodo dragon will be screened. They are more likely to be different compared to other lizards. Living without suffering infection in bacteria-rich environments reflects rapid evolution of immunity-related genes along with expansion of immune gene families and antimicrobial peptides. In fact, in a previous study, it was found that 48 novel potential cationic antimicrobial peptides in Komodo dragon might be responsible for their antimicrobial resistance. To carry out the comparison of the different genomes, firstly the genes will be compared to determine the possible synteny between the different species. Both macrosynteny, synteny involving a large portion of the genome, and microsynteny, involving only a few genes, are going to be studied. Comparing the genomes of the species selected, orthologous regions and the prediction of the location of some of the genes in Varanus komodoensis can be seen. Moreover, their structural differences due to chromosomal rearrangements are going to be analyzed given the fact that they might have a role in the antimicrobial resistance. SynMap and SynMap 3D from the Koji platform will be used to create dot-plots to search for macrosynteny between the varanids, and it would be possible to detect inversions, translocations, transpositions, duplications, or deletions. Microsynteny will also be visualized with the Jibo tool from Koji also, a tool designed to visually compare genomic regions using both local and global alignment algorithms. Gene duplication may have caused neofunctionalization, obtention of a new function, subfunctionalization, so the function becomes more specialized, or higher dosage, so there are more copies of the gene present, and that entails faster and more intense action. However, in most cases, gene duplication will have no effect whatsoever because the obtained copy will probably become a pseudogene with no function. Looking at the synteny, it will also be detected whether there has been a fusion of chromosomes through evolution. If it were the case, the genes involved in the fusion zone will present important changes. Taking into account the synteny results, the genes or genome sections that present structural differences will be further analyzed because they are candidates to be responsible for antimicrobial resistance. Positive selection refers to the process that appears as a consequence of adaptive evolution, to respond to a change in the conditions of the environment and survive. Positive selection might be the cause for the differences between the Komodo and the other closely related varanids, triggering mutations that can lead to the appearance of new genes with new or specialized functions or gene loss. Since Komodo dragons live only in the Komodo Island and other surrounding ones, positive selection may have acted up in evolution and caused speciation because of the geographical separation of Komodo dragons with other varanids, developing antimicrobial resistant genes as an adaptive response to their new habitat confined in an island. Orthologs that present mutations and other genes that appear either only in the Komodo or not in the other varanids, or that appear in both varanids but not in the Komodo, will be studied. The main focus will be the gain or loss of certain genes. First, in order to identify homologous genes and find one-to-one orthologs between these varanids, the orthologous matrix pipeline OMA will be used. This pipeline clusters homologous sequences from complete genomes in order to identify orthologous pairs of sequences and infer HOGs, hierarchical orthologous groups, which are groups of genes that have descended from a common ancestral gene in an specified taxonomic range. The tool SynFind from Koji that identifies homologous gene pairs using sequence similarity will also be used in order to check the results obtained. The effect of positive selection will be calculated thanks to the Ka-Ks ratio, or non-synonymous synonymous substitutions ratio, which takes into account which substitutions have been silent or not. If this ratio is bigger than 1, that indicates that the analyzed zone is under positive selection. All the genes that are under positive selection will be further analyzed, studying how has their function changed. The most likely option would be that with the new mutations, the gene or genes have obtained a new function, but it is also possible that it has produced a pseudogene and the lack of that gene in Komodo is what makes it resistant. Gene families are groups of genes with similar sequence that contribute to the same overall function and were originated from one original gene. Expansion of gene families is associated with gene gain, duplication, while contraction is associated with gene loss. These two entail a series of consequences that were mentioned previously. Usually, a higher amount of genes dedicated to the same general function with the same or different specialization means that function is highly relevant to adapt. Higher number of genes would imply higher immunity or resistance. Duplications of a certain gene in the Komodo dragon would generate paralogs of that gene, expanding that family. Also, as mentioned with gene loss and pseudogenes before, the contraction of a gene family could be responsible for antimicrobial resistance if the reduction of genes entails a lesser effect from bacteria. In order to detect expansions and contractions of gene families, the software CAFE3 will be used to analyze the HOG data obtained before from the OMA pipeline. This software will measure the size of the different gene families so that it can be compared among the different varanids. The families that have changed their size will be further studied because they are candidates to be responsible for antimicrobial resistance. As mentioned before, around 48 novel cationic antimicrobial peptides, all of them except for one derived from histone proteins, have been linked with antimicrobial resistance in Komodo dragon, and have been proven effective against bacteria such as Pseudomonas aeruginosa or Staphylococcus aureus, both present in Komodo dragon saliva. These antimicrobial peptides play an important role in immune response. Knowing that these peptides are of special interest since they have been linked before with antimicrobial resistance, the genes that codify them will be studied with more detail, looking for signs of expansion or contraction of their gene families or action of positive selection. It will be studied whether these genes are present in Komodo but not in the other varanids, if there is more or less amount of copies, or if they show any differences in their sequence. As for the economic considerations of this experiment, the cost of this project ascends approximately to €100,000. It must be also taken into account that the Illumina and BioNano prices that appear in this table correspond to the sum of all three sequencing projects from the three varanids used in the experiment. To sum up, with the comparative genomics approach taken in this study, we will be able to detect the potential candidates responsible for the antimicrobial resistance of Komodo dragon. These potential candidates will be gene families that present expansion or contraction, genes that are under positive selection, or genes or parts of chromosomes that present structural changes. The aim of this study was to find potential genes involved in the antimicrobial resistance mechanism that confers Komodo dragons the ability to survive despite having pathogenic bacteria living in their mouths. In addition to this comparative genomics approach, other supplementary studies should be done in order to obtain better results, such as a metagenomic study of the bacteria living in their mouths. The results obtained in this study could have a beneficial impact in medicine, since bacteria are the most frequent cause of infection in humans and other organisms. Knowing how Komodo dragons fight against bacteria could help to develop new therapeutic treatments to fight bacteria in other organisms.
Thank you for your attention.